CRISPR, short for clustered regularly interspaced short palindromic repeats, has revolutionized genetic engineering. This groundbreaking technology allows scientists to edit DNA with unprecedented precision. Discovered in bacteria as a defense mechanism against viruses, CRISPR has been adapted for use in various organisms, including humans. Its potential to correct genetic disorders and improve human health is immense. Unlike previous gene editing tools, CRISPR can target specific DNA sequences and make precise cuts. This opens up possibilities for treating genetic diseases that were once thought incurable. However, with great power comes great responsibility. The ability to alter human DNA raises significant ethical questions. CRISPR's medical potential is astounding. It offers the promise of curing genetic disorders by directly correcting faulty genes. This could transform the lives of millions of people suffering from hereditary diseases.
Researchers have made significant strides in using CRISPR to treat beta thalassemia and sickle cell disease.
CRISPR could also be employed to develop new treatments for HIV by targeting and disabling the virus's DNA within infected cells.
Despite these promising developments, CRISPR also raises profound ethical concerns. One of the most contentious issues is the possibility of creating designer babies. This refers to the use of gene editing to select or enhance traits such as intelligence, physical appearance, or athletic ability. Another ethical dilemma involves germline editing, which changes the DNA in sperm, eggs, or embryos. These changes are heritable and would be passed on to future generations. The societal reactions to CRISPR are mixed. On one hand, there is excitement about the potential to eradicate genetic diseases and improve human health.
On the other hand, there are significant fears and concerns. One major fear is the potential for increased social inequality. If gene editing becomes a tool for enhancing human abilities, it could create a divide between those who can afford such enhancements and those who cannot.
There are also concerns about the unintended consequences of gene editing. The long-term effects of altering human DNA are still unknown. Additionally, there is the worry that CRISPR could be used for malicious purposes, such as creating bioweapons. Public opinion on CRISPR is shaped by these fears and hopes.
Given the profound implications of CRISPR, robust regulations and oversight are crucial. Governments, scientific communities, and ethical bodies must work together to establish clear guidelines for the use of gene editing.
These regulations should aim to maximize the benefits of CRISPR while minimizing its risks.
Rigorous testing and clinical trials are necessary to determine the potential side effects and long-term impacts of gene editing.
Another important aspect is addressing the ethical concerns associated with CRISPR. Guidelines should be established to prevent the misuse of gene editing for non-medical purposes, such as creating designer babies.
There should also be restrictions on germline editing to prevent heritable changes that could affect future generations. International cooperation is vital in regulating CRISPR.
The global nature of scientific research means that regulations must be consistent across countries to prevent unethical practices. Public engagement is also essential in the regulatory process.
CRISPR represents a monumental leap forward in genetic engineering. Its potential to cure genetic disorders and improve human health is unparalleled.
Balancing the benefits of CRISPR with its risks requires careful consideration and responsible oversight.
It is crucial to prevent the misuse of gene editing while ensuring that its benefits are accessible to all.
Society must navigate the ethical crossroads of genetic engineering with caution and foresight.
By fostering open discussions and considering diverse perspectives, we can ensure that this technology is used for the greater good. In conclusion, CRISPR holds the promise of transforming medicine and improving human health.
By balancing progress with ethical considerations, we can harness the power of CRISPR responsibly and ensure that its benefits are realized for generations to come.